Next up, let's look at how proteins are chemically digested, and this again principally takes place in the small intestine, but there is a little bit of it that happens before that in the stomach. So let's start out at the stomach and look at what exactly happens to help digest proteins in the stomach. Now there are a number, before I talk about the stomach in general, let me just give you a couple of terms here. One is proteases. You'll sometimes see textbooks use this term. Proteases is just another name for peptidases, and peptidases are simply enzymes that have the ability to break down peptides or proteins. There are a number of these proteases or peptidases that are manufactured initially as zymogens. And you'll remember zymogens are those types of enzymes that are in an inactive form. And in order to become active, they usually have to be cut up into somewhat smaller pieces. So let me give you an example. You remember that you have the chief cells in the stomach, and they manufacture pepsinogen. You can tell that pepsinogen is a type of zymogen because it ends in OGEN. And this pepsinogen, as you can see down here in this picture, consists of a long chain of little amino acids, and they're representing those amino acids with these little spheres or little balls. You know, in the presence of hydrochloric acid, we can convert pepsinogen into its active form, into pepsin, in other words. And that happens because hydrochloric acid has the ability to cut apart this pepsinogen. And we get a little bit of peptide that gets removed, and now we have the active form of the enzyme called pepsin. Again, it's a shorter version. So you can see with zymogens, we're chopping something off, creating a shorter molecule that becomes the active enzyme. And the way that this pepsinogen, or pepsin I should say, works is that it then goes out and it encounters polypeptides. And any place on that polypeptide chain where I have a tyrosine, that's one of the amino acids, immediately adjacent to a phenylalanine, that pepsin breaks apart the bond. So it goes along all the polypeptide chains. Any place there's a tyrosine, phenylalanine side by side, it chops them apart right between them. And so what this does is effectively take our polypeptides, and in the presence of water, because remember this is hydrolysis, we're using water to split it apart, and in the presence of pepsin, we are manufacturing a bunch of oligopeptides. I'm still thinking about sugars. So that's the first step. This happens in the stomach. Remember, the stomach has a pH that's relatively acidic, so pepsin works best in a very low pH environment. Now compare that with what happens in the small intestine. In the small intestine, the pH is more around, somewhere around 9 or so. So it's much more alkaline in nature. So the enzymes that are at work in the small intestine, in its lumen and on the brush border, are more responsive to an alkaline environment. So you'll remember again, the first order of business when that chyme entered in the small intestine was to get the pancreas to release a lot of bicarbonate ions to neutralize that acidic chyme from the stomach. And if you're interested, the process by which those bicarbonate ions are made in the small intestine is pretty much the same idea as we use for making hydrochloric acid in the stomach. The only difference is that the hydrogen ions go into the blood and the bicarbonate ions go into the small intestine, into the cells that are making the bicarbonate. So we're just kind of reversing the process to make bicarbonate. So rather than the bicarbonate ions going into the blood, they eventually get manufactured by the pancreas and released into the lumen of the small intestine. Okay, what are the different types of enzymes involved in protein digestion? Let's take the lumen of the small intestine first. In the lumen of the small intestine, we receive a number of different enzymes from the pancreas. So let's look at the different types of enzymes the pancreas makes. Pancreas makes an inactive version of a protein enzyme called trypsinogen. And there's that OGEN ending, tells you it's a zymogen. Pancreas also makes chymotrypsinogen, also in an inactive form. Those inactive versions are released into the lumen of the small intestine, and they bump up against a brush border enzyme called enterokinase. Okay, very important. This enterokinase is on the brush border. It is actually part of the intestinal wall. It is situated on those microvilli. When chymotrypsinogen and trypsinogen come in contact with enterokinase, the enterokinase chops them apart and makes smaller active versions of those enzymes, which are called trypsin and chymotrypsin. So we've now got active trypsin, we've got active chymotrypsin that's floating around in the lumen of the small intestine, and as it comes into contact with both polypeptides and oligopeptides, it breaks them apart into even shorter oligopeptides. Okay, so here we go. We've got some oligopeptides and polypeptides here, and we're going to convert them into even shorter oligopeptides that maybe are 8 or 9 amino acids long, or 4 or 5 amino acids long, something like that. Again, remember, we have to consume water in this process because this is hydrolysis. So we're hydrolyzing these into even shorter oligopeptides. The pancreas also makes one other enzyme called procarboxypeptidase. Pro means that it's in its inactive form also. This gets converted into its active form, which is carboxypeptidase. And that too helps to chop up polysaccharides and oligosaccharides into even shorter oligosaccharides. All of this is happening in the lumen of the small intestine as a result of enzymes that were manufactured in the pancreas and then released into the lumen of the small intestine. Now we've got a bunch of oligopeptides, and we got to get those oligopeptides broken apart into individual amino acids. The process by which we do that occurs on the brush border enzymes. Okay, so now we're not looking at what's happening in the lumen anymore. Now we're looking at what's happening on the intestinal wall, on those microvilli. Here we are down at the microvilli, and there are a number of enzymes that are installed right on those microvilli. Their job is to actually break apart these short little oligopeptides into individual amino acids. Now, let me remind you of something. The generalized structure of an amino acid is a central carbon with a hydrogen, an amine group, a carboxyl group, and then whatever R group to make it whatever amino acid it is. Okay, so if I look at an individual oligopeptide like this one right here, on one end the oligopeptide ends in the amine group of that final amino acid. On the other end, it ends in the carboxyl group. Well, there are certain brush border enzymes that specialize in removing different amino acids off of one side or the other of that oligopeptide. So, for example, we have something called carboxypeptidase. What carboxypeptidase does is it looks at the carboxyl end of an oligopeptide, and it begins to hydrolyze or remove off one by one amino acids starting on the carboxy side of the chain. Now notice carboxypeptidase was an enzyme that the pancreas also made. So this is one of the few enzymes you're going to learn about that's found in 2 locations. It's manufactured by the pancreas in an inactive form called procarboxypeptidase, and it's released in the lumen where it becomes active. It is also present already in its active form installed on the brush border. So we see it in 2 locations. What about on the other end of an oligopeptide? On the other end of the oligopeptide, we end in the amine group. Well, there's an enzyme that is good at taking off amino acids from the amine side of the oligopeptide, and that is called aminopeptidase. Again, also on the brush border. It's actually physically attached to the microvilli. So these guys will, the carboxypeptidase and the aminopeptidase, they will be working at these oligopeptides, taking off one by one amino acids off of either end. Remember, this is all contact digestion. That means I actually have to get those oligopeptides to bump up against the microvilli where those brush border enzymes are located in order for a hydrolyzation event to occur. Now, eventually, at some point, we're going to get to where there are only two amino acids connected together left. In other words, we have a dipeptide. And at this point, well, geez, we have one ending as an amino group and the other end is a carboxyl group. It turns out neither the carboxypeptidase nor the aminopeptidase can handle one of these two amino acid long chains. We have to have a third enzyme here called dipeptidase. Its job is to do the final breaking of the joining between the final two amino acids. Again, this is all contact digestion. It's happening at the microvilli. We're making individual amino acids at the microvilli, and that's convenient because the very next thing we want to do is right there at the microvilli, we want to absorb those amino acids and get them into the blood. So conveniently, as soon as contact digestion occurs, we can readily absorb those amino acids. And we do that using protein channels that are similar to the ones that we use for glucose. We rely on the high concentration gradient of sodium out in the lumen here. We allow sodium to move down its concentration gradient, and along with it, it brings an amino acid with it through that special type of protein channel called a sodium protein channel, or sometimes called a sodium-dependent amino acid transport protein. It's very similar to the sodium glucose transport proteins. Same idea, but instead of moving glucose, we're moving amino acids and getting them into the actual blood. Okay, so that's a process by which we chemically digest and absorb proteins. The next two types of organic molecules we'll talk about are the final two. Those include lipids and nucleic acids.